So let's start off by sharing some bits. And if I can, we want to go to a window and want to go to emails. I can close down another document. Let me just see. Yep. That's more stuff closed. And now I want to go to emails. There they are. And share. And go over the correspondence. I might have missed one, but I should have most of them.
We've got a few this time.
Yeah, I got a few came through. Colin sent a picture of Syagris, and there's another one where he actually has the image present. No, there it is.
Mm-hmm.
Okay, so is that clear enough for you guys?
Yep.
Okay, great. So we've got a disease issue, which is precisely what I want. I'm just going to make it a little bit smaller. Let's see if I can get that even bigger still. No, that's going to be it. So we've got a tree, a palm. A palm's a tree by definition. Anything that's woody that can beat a person to death makes it a tree. So we've got a tree that's stressing back. What's the first thing you notice?
The underside, so the lowest section of the fronds seem to be dead more than the upper section.
So we're getting some bits dying off here, yeah. What sort of disease does it look like?
Virus.
Could be a virus, yeah, except viruses tend to form along distinct lines. That is, the virus, once the virus is told to stop communicating, we get crisp lines around the vascular system. So whilst it does look a little bit virusy, what we can be sure of, it's not a macroorganism. That is, it's a microorganism. It's something that's small. And viruses, for the sake of it, aren't living, but we'll treat them like they are living things. So if it's not a virus, and it could be, what else might it be?
Fungal.
Fungal. And there's some indicators on it that suggest that it's fungal by the way that the margins seem to be progressing. So that would be the most common thing that I'd expect to see. And so, yes, we've got a Syagris that's dying off now. Is Colin online at all?
Not sure.
It was Colin that asked it. Yep.
Yeah.
So this is a possible nutrient deficiency. So there are a number of things that could lead to this, but it's unclear. What would we do to work it out diagnostically?
Get a lab report.
Yeah, so send it off and see whether they can culture. Just as a heads up, and it's unlikely to be, but there's a new form of Fusarium oxysporum FS, form of species palmae, and it's affecting pretty well all species of palms. And we might not think that as being something that's big or important, but Fusarium is a particularly nasty organism. Am I still there?
Yeah, it's not 100%, but it's okay.
Let me see what happens if I turn off. I can't turn off my. Oh yeah, I can. Camera. I can turn off my camera. That might make it a little bit better. How's that? Is that any better?
Yeah, okay so far.
Yeah, okay.
Intermittently, so I'm not sure yet.
So Fusarium is a nasty thing. Fusarium was the thing that caused the Great Potato Famine, and Fusarium affects bananas. It's a form of Fusarium, a Panama disease that kills Cavendish bananas, and we have as our primary banana crop in Australia Cavendish bananas. And so we always need to be cautious when we see things that
Panama affected ladyfingers mostly.
I could be wrong. You can double check me.
I think, I'm relying on memory, but my memory is that, ladyfingers are the ones, and to the extent where some areas no longer grow ladyfingers, and one of the reasons is that the ordinary banana varieties are popular, is that they're not susceptible to that.
Just looking up Panama disease and Fusarium bananas, Cavendish. Panama disease, Cavendish are susceptible to Panama TR4.
They're certainly not as susceptible as the ladyfingers, that's for sure.
That's quite possible, yeah.
Yeah, that just absolutely hammers the ladyfingers to the extent where some growing regions just don't produce any anymore.
Yeah, it might be strange to talk about bananas, but it's actually a topic that was raised several times in the correspondence this month. So we will talk about bananas. But yes, look, these diseases are particularly nasty. So Fusarium is one that's difficult to deal with. Interestingly, there is a cure for Panama disease developed by an Australian. It involves splicing a resistant gene from another banana into the Cavendish gene, which you can do. Plants generate rather than regenerate, so it makes it a little easier to play that game. But no one will accept it because it's modified. Well, of course, all it is modified by a length of banana gene that's already in bananas. So
doing a lot of work on trying to develop disease resistance in bananas, obviously for good reason. And they have come up with some new varieties recently. So I'm thinking particularly of gold finger maybe. And, it's tough, grows well, disease resistant. It's not popular because it doesn't taste any good.
Yeah, and that's a real issue. Is Cavendish is a taste that we've acquired. I mean, if you've had, there are lots of different varieties of bananas in the world, and Cavendish was selected and bred. It's for its taste and its color and its shape and size. So it's a pretty neat little banana in that regard.
So what do the banana growers do to control it at this stage?
That's a good question. Where it's a pretty major disease overseas, and remembering that Australia is only a small grower, so South America is by far the largest. Once you've got Panama identified, you'll kill off the host plant. You'll kill off plants around to reduce the likelihood of spread. You'll cover over the ground where the infection started, and you'll isolate a large block out. A large block, maybe 200-300 square meters of production land is isolated out. Overseas, most banana production has high sterile environments, so boots, washers, walkways, very limited access because it's fairly easily spread. These soil-borne pathogens, and remember, it's not a minor thing like Phytophthora, which again is easily spread, but it's a primary pathogen. That is, it tends to kill quite readily, and it breeds and alters its genome fairly effectively as well. So we're going to continue to find for the rest of our lives and for many generations to come, I suspect, varieties of casts of Fusarium that are going to kill off plants, and that's a real issue. So we do have, as you probably already know, Fusarium oxysporum canariensis. That's the one that's doing the Canary Island palms. I'm unsure whether albanensis is here. That's the one that kills the true date palm. That's where we started to worry about it in palms. We've certainly got Fusarium, I think it's Fusarium solanaceae, that's the one that does potatoes and tomatoes. So these diseases are fairly broad spread, and this particular genus Fusarium is nasty in most cases. In fact, Polyphagus shothole borer, have we talked about that yet?
I'm not sure. It seems to be an ongoing situation, so I'm not
Okay,
clear on the latest.
let's raise it. Who's here from Western Australia? Nobody. There's some dirty people from Western Australia that have brought in this disease called Polyphagus shothole borer. It's a little Curculionidae, a weevil. And weevils have these little things on their bodies called mycangia, and they're made for transporting fungi. So this, and not uncommon in beetles. These beetles are going in, they're boring little holes in trees, particularly trees with low levels of what we call extractives or heartwood that's very light. They're really the preferred option. And so these Polyphagus shothole borers come in. Poly means many, phagus means to eat, so it eats many different things, broad range of host species. It's been introduced in Western Australia and it's killing off poplars, maples, rhododendrons, doing some damage to liquid ambers, damaging figs. And to try and control it, for example, the Western Australia government just recently, I think, we're removing 70 or 80 heritage fig trees to try to stop the spread of the disease. So that's a pretty serious situation. And these things are difficult. When we get these disease outbreaks in plant material in Australia, historically we're worried about it. So we all worried about what was going to happen with myrtle rust. And sure, a couple of species have been really affected, but most species have handled it fairly well and it hasn't been as big a problem as we thought. Chevaephus celti came into the country. Boy, it just knocked things around, but it seems to have come to a halt. It's there, but it's not doing much. And I think a part of what we're seeing is natural predators, natural parasitoids, things that kill the host, are starting to keep things under some form of balance. So the first 3 or 4 years of Chevaephus celti, we treated every spring on a whole pile of trees. Nowadays, we don't even get a call. I'm sure it's there. I've seen it, but it's not at the levels. We used to be able to see, 1/4 of a millimetre of sap underneath a celtis in springtime because there was that much sap being pushed out. Now, you can park under a celtis and not be worried about getting too much sooty mold on your car. So, yeah, these problems are a problem. Polyphagus shothole borer is a problem because you can be almost certain if it got to Western Australia, it can get to the East Coast. And when it gets to the East Coast, that's going to be a problem, particularly when you get down to Melbourne, because Melbourne's got a lot of these lovely trees like maples, done really well with maples. They look great, but they are just absolutely hammered. Maples are species, and you think about it, you see decay in maple branches. They'll often get a bit of heat damage or something, a big wound, and then you see them become C-shaped branches because all of that wood decays quite rapidly. So what happens is the beetle introduces fusarium, and it's the fusarium that's doing the damage, not the beetle. Fusarium causes a wilt, the tips die back, the tree starts to lose all vigor and it's gone. If you go into a branch, that's 100 mils in diameter, everything from there on in is gone. Within a matter of a few years, the tree's dead, lost all vigor and gone. So pretty nasty disease. So something that we need to keep an eye open for. There's some really good information online. Grab some and have a read. Might see, Peter, whether we can get someone from WA to do a presentation on it, although there's been some tensions there about how that process is working. It would be great to at least have some more information provided on that.
It would be good to get an update on how things are travelling, because I think the situation seems to be changing a bit. And as you say, a lot of these diseases make a mess when they first come in, but they settle down. I'd be interested to know how they're dealing with it overseas, because obviously that's where it comes from, and they have trees left, so they must be dealing with it somehow.
Yeah, look, and again, the part of the reason why these things are often problematic is the natural predators aren't there. And so it depends on what else is there to step up and take that place. And that's what's really going to determine how these things perform in the longer term. So yes, look, I would suggest two things are possible. One is taking a sample of the soil to see what's happening there. And the second one is taking a piece of the foliage at the margin of the diseased and the non-diseased. So an area like here, the end, the disease is essentially gone. So we want to be closer to that line, and maybe 4 or 5 of those sent off to a pathology lab to have an identification done, and a sample of the soil sent off for checking. And there are other things that could have caused that. One of the things I do notice is that at least one side near the gutter's gone, so it could potentially have been a lightning strike and arc out onto the guttering system. So there's another possibility, but certainly something's having a go at the foliage now.
Can I just ask a different question?
Yeah, you just did. Do you want to ask another one, Rob?
No, I don't really understand the vascular system of a palm tree. Can you talk a bit about that?
Yeah, great. So palms run, they're monocot, so monocotyledonous plants. As a rule, no cambium. There are a couple of exceptions to that, but palms certainly don't have a cambium. And they lay tissue down in vascular bundles. And a vascular bundle is an area that's enclosed by a sheath that isolates it, that keeps it as a solitary unit. Xylem and phloem cells, so cells for transporting up vessels that are made up of vessel elements, and phloem cells for transport of sugars, and so they are upwards and downwards in direction, so bidirectional transport in those. And they run all the way up and down, and then around those there's a scientific term for it. It's craplodes. There are craplodes of parenchyma, and they may be living or they may be dead depending on the species. So something like Cyagrus, which is what we've got here, lots of those cells are alive. So parenchyma is fairly long lived in those.
Can I just ask, the description of these vascular bundles seemed to me to indicate they were always just ran up and down, but I'm not sure that's the case.
No, they don't just run up and down, because the trunk's not laid down just up and down. It's actually laid down at an apex and it's pushed outwards. So they go up and down and then curve towards the outside edges. And if you look at a palm, you can actually see the vascular bundles going to the PDL bases. They're really visible. So
that systems might sound pretty simple and straightforward, but it's a bit more complicated than that.
It's extremely complex in the way that it interacts, yes. And there's a gentleman called Zimmerman that did a great movie of it. I have it somewhere, where you can see the way that the vessels are moving, and they do move, not move within themselves, but geospatially as you go up the tree or up the palm. So
does each frond have its own vascular bundle?
There are vascular bundles going along in each frond. Let me see. I don't know that I can do one, but let's just see if I can. If I can find one, I'll show you. RHACHIS. really odd sitting in an office typing in the dark.
Well, you can turn your light on again. We're not seeing.
Oh yeah, I can too. Thank you, Peter. Boy, not that bright in this end of the world. No, I thought I might have been able to find one quickly. I can't. I don't want to spend too much time. But yes, they've got lots of vascular bundles going up and down in the palm leaf as well, in the frond.
So there'll be no rays.
No. So the difference with monoclots is there are no rays, and I have found a shot that will probably do. Okay, I've got to go back and stop sharing. I should be able to share now, Peter, my entire screen because I'm not broadcasting me. So let's see if I can do that. Entire screen.
Your microphone's off, Peter.
I don't know whether it'll allow me to do it. Yeah. Can you see an image here on the right-hand side of my screen, guys?
No.
No. Okay, so it won't let me do it. Oh, yeah. Yeah, it says that it is. Okay. Can you see this? A purple screen, a purple image now.
Yep.
Yep.
Got it.
Great. Okay. Well, this is a rachis of an oil palm, and these are all vascular bundles here. These things that have got this dark area around them, and out here too. That's another one, and out here and out here. Lots and lots of vascular bundles, just huge amounts. And this is what we're looking at. We've got phloem, we've got metaxylem, major xylem. So we've got the xylem cells here, and we've got a sheath around the outside edge, and some parenchyma in there. And then the same, we've got parenchyma in between all of this. So this is a parenchyma that you're following around with my finger. So now that's nice and ordered. And it's nice and ordered there because there's not too much going off to the side at this point. Where you get a leaf or a leaflet come in, it gets a little bit more complex. So hopefully that gives you an idea. It's not as simple or as easy. And at some stage or other, I encourage you to all grab yourself a microscope and have a look underneath a microscope at a thin section. You can cut this with a razor blade or a box knife, Stanley knife, call them whatever you want, and look at it even under a loop. Now if I go to a palm stem, and this is a palm stem again, you can see the vascular bundles, lots of parenchyma, huge amounts. And here it starts to get a little bit more indiscreet, because here we've got little bundles rather than big bundles, and they will have connected up at some stage or other to the petiole base at the base of it. And this is a neat little illustration that again shows you some of that complexity where things cross over to connect up to those spots. Does that make a bit more sense for you, Rob?
Yeah, that's good. Thank you.
It's a mess when you look at it, but it's a very, very ordered and intelligent and deliberate mess. So it's not just random, it's actually serving the purpose. And it's more easy to see some of these things in palms that have got very big petiole bases than something like a Washingtonia, which has got a very discrete base on it. So hopefully that makes some sense. Okay, so, oops. Yep, I'm sharing my screen. We're going back. Any other questions on the palm whilst we're at it? I will say something else, Rob, because you've raised the question. There are no rays. And the question that it raises is, do palms compartmentalize? What do you think?
Yes.
I think maybe they do.
Yeah, in fact, they
Hmm.
do. What they don't have is wall 2 or wall 4.
Mm.
So they still have wall 1 and wall 3. So wall 1's weak. So decay in palms tends to spread up and down quite easily. And then it radiates out evenly around the infection point past that. And if you come along to an anatomy workshop at some stage or other, we'll show you what the palm does in terms of dealing with it. But yes, they do compartmentalize. 2 walls, not 4. Why 2? Transport in along the medullary rays is one of the ways that decay spread. No medullary rays or no of radial parenchyma, so there's nothing going in and out. That's gone. And no wall 4 because there's no cambium, so there's nothing being produced on the outside. And sometimes people look at these things and say, oh, look, they're an inferior plant. But in fact, they produce this one outer periderm bark, effectively, that lasts the entire length of the life of the palm. And on occasions, that can be several hundred years, well, close enough to several hundred years, couple hundred years.
Mike, did you say that there was parenchyma in the palms, though?
Sorry?
Did you say there was parenchyma?
Oh, lots of parenchyma.
So how does that get organised?
That's parenchyma tends to be more just random cells, but I'll show you again if I can go to that last one that I had. I'll just go to history and pull it up. So this is parenchyma here, for example, and this is parenchyma here, and parenchyma here.
So is it running sort of?
Yeah, and in many cases it's long-lived. So it's adjacent to the vascular bundles, holds the vascular bundles in place. So it's one of the reasons why a lot of monocots are quite easy to mess with. Of course, not all of them are easy to mess with, but you know, they do some weird things. So, for example, you can, and I think I've talked about this before, you can propagate palms by effectively cutting off a stem and putting it in the ground. And that shouldn't surprise you if you think about date production, because all date production is done using off shoots. So let me see if I can show you a picture of that date. That's not true either. A guy called Zayed figured out and mastered the process of tissue culture about 15, 20 years ago. And so he is using tissue culture vegetation. But they take these little offshoots here at the base of the palm and cut them off. And oftentimes with no roots on them at all, they're just pulled off and they're allowed to do their merry way. So pretty neat when you think about it. So they've got this thing called a root initiation zone, and that root initiation zone on Phoenix, for example, Washingtonia, lots of other things, runs all the way up the palm. So again, you know, here you've got a high offshoot. You can take that off, plant it, and grow a new one. And I guarantee you there's no roots on that. Freaky? Doesn't make sense because we're all told that, of course, you can't do that. Palms have a single meristem, but the root initiation zone is one that goes all the way up. And this is another one that I was talking about recently with some people. The palm likes to be into the ground a fair way, and so it starts its life with contractile roots, the roots that pull the meristematic point at one stage where it's producing everything. So it's down here. It pulls that down to the ground rather than being high. So that makes it a really interesting plant in many ways. So we talk about them as being simple, and I guess they are, but they are also complex and quite well evolved. So amazing.
So how can you get a shoot coming off the trunk up the top there when there's no cambium?
Because you've got parenchyma, and parenchyma is totipotent. The word totipotent, in simple words, means can do whatever the hell it wants. So you can get roots to grow off a tree when there's no roots there by just wounding the tree, putting a little bit of peat moss around it, a typical aerial layer, and it'll grow roots. Because the parenchyma got the message. It's dark, it's wet. You could be whatever you want to be. The pressure's off you. The adjacent bark's gone. For goodness sakes, form a bit of callus, which is the first thing. Callus resets the plumbing of the tree. It allows it to deal with injury. So it resets the callus, and then off it goes. It's reproducing quite rapidly after that. And it can produce roots. It can produce new shoots. It can do all sorts of wonderful things. And we get this view, and rightfully so, that trees are often quite simple, but at the same time we miss in that the view that trees are also significantly more complex in other ways. So chemically they're more complex, organ simple chemical complexity, but they do things that just don't make sense. Like if you cut off a fig root, put in a barrier, the fig root will go around the barrier back to the original spot and want to keep growing as though the barrier wasn't there. Now that's freaky. That infers, that it has some sense of navigational skills, which is not what really is happening, but it gives that appearance.
It sounds like it's using Google Maps.
Yeah, and there's a creeper in South America that actually changes its leaf based on the host that it is growing on. So it actually engages in mimicry, almost like it's got eyes. Well, they don't have eyes, but they've got photoreceptors. Well, what's an eye? An eye is just a highly ordered photoreceptor process in our case, in two spots. That doesn't make what the plant does any less significant. It's just that it's different. And we tend to underplay what plants do, not saying they have feelings, but they can respond to stimuli, why don't they have feelings? Because they don't have a central nervous system. Why does the nervous system have to be centralized? Why can't it be dispersed? And so these arguments are being carried on regularly. Particularly, there's an institute out of Italy and one out of Japan that are looking at these behavioral patterns of plants, and they really are amazing pieces of research that show things like plants have memory. Trees can remember things, not in the same way we remember, but they can still store information. They know, for example, that they've had that disease before. They can recognize that insect, and that's pretty clever. Tobacco is another one that I love talking about. Tobacco is a member of the Solanaceae group. Tomato family, potato family, and tobacco is pollinated at night by a hawk moth. The hawk moth feeds on it as well. And if the population of the hawk moth gets too high, the plant stops night flowering and produces a day flower instead. That's slightly different, that it's not pollinated by hawk moths, and at the same time puts out a chemical, a pheromone, a scent attractant that attracts the wasp that predates on the hawk moth larvae. So it actually puts out a scent that says, hey, all you wasps around that like hawk moth larvae, here they are, they're on me now at a high level. That's a level of planning and thinking in human terms that's quite advanced. In terms of plants, it's just some really good, clever biochemistry. And whilst I say that, I've got to be fair, our thinking is just really clever biochemistry. So it's just a different sort of biochemistry. So I don't want to downplay plants, but I don't want to make them sound like they're humanoid at all. They're not. Very different, but they're sentient, for example. They respond to stimuli and touch. We know that. And they do that in lots of different ways. So yeah, pretty freaky things. Okay, let's go back to some pictures and some questions. A great question. Peter Gray says, how come bananas, yellow watermelon, even pineapples do not have any seeds, or at least visible seeds? Well, Peter, I'm outside of the field of expertise of watermelons and pineapples, but I am somewhat familiar with Musa bananas. Probably not quite as good as you, but Cavendish that we deal with, that's the most common banana we eat, is a triploid banana. The watermelon that you talk about is a triploid watermelon. And when you've got 3 sets of chromosomes, that's what triploidicity is, and you go to split them in 2, you realize that they don't split into 2. And so we get sterility, and that's a problem and an advantage. So there are people, for example, that crossed wheat and barley. Wheat and barley forms a plant called triticale. And triticale is, by all intents and purposes, sterile, except in the laboratory, the nice plant scientists that understood a little bit to do with genetics and bits and pieces kept on working it until they found and developed a non-sterile form. And triploidicity is a really neat little thing to look at. Birthday candles, Banksia birthday candles are triploid Banksia, bigger fruit. So things that are triploid are often increased in size. And if you think about a Cavendish banana versus a ladyfinger, that sort of makes some sense. The Cavendish is definitely a bigger fruit. So triploidicity is a way of achieving that. And it comes with the advantage of being sterile. Colchicine, which is a chemical used for gout in people, is a chemical that's been used to artificially induce triploidicity. There are other things that have been used to do it as well. And hybrid breeding. So breeding out of a genera. Even breeding at extreme ends of a genus may get you a sterile form. That's one of the things that we've done to produce things that aren't going to be a problem. Now, up in Peter's end of the world, there's this beautiful tree called Cinnamonum camphora. Absolutely exquisite tree, but it produces tons of fruit. And if you could get a sterile form of that, it would be a great tree. So that's something that, if we really are looking for developing things, we're going to start looking at that. And we've, in fact, done that. And we're seeing lots of sterile forms. There's sterile Acer negundo out there that are used. There's sterile Melias. There's 2 sterile cultivars of Melia Zeterac now. So we will see more and more of that. There are some advantages.
So is Parthenocarpy all encompassing these various ways of getting there?
I don't know the answer to that, Peter. That's a good question. I think it covers at least some of the ways of getting there. So bananas do actually have seeds. Lots of species
They're just not viable.
Oh, no, viable seeds. You can grow ladyfingers. I'm pretty sure the seed is viable.
Oh, okay.
Okay, but Cavendish is sterile because it's triploid. And what that means is every Cavendish is an offshoot. We grow them from offshoots. And that means that clonal everything's the same. And clonal things have some real advantages, but they also have some horrendously horrible disadvantages. And we're starting to see that now in Sydney. The Buprestidae beetle, the jewel beetle, Vitusa, I think, Nathaniel, you remember seeing it. I think we bred some in class at one stage. I think you were still around those days. That little beetle has decided that Tristaniopsis lorina luscious is a desirable species to kill. And yes, Nat, you can discuss the images if I can find the images from last time round. You can resend them to me and I'll check in a few minutes if you'd like. So yes, it's decided to take on Cascade. Now the drawback of that is as these things start to build up numbers, and you've got lots of Tristaniopsis luscious out there, the beetle numbers get high. And as the beetle numbers get high, the damage they do and the extent of the damage increases as well, because they're going to come out and the first thing they look for is something that's vulnerable, and that's going to be the Tristaniopsis luscious nearby. So this mass planting is problematic. Most of you will be familiar with an European tree, Pinus halepensis, the lone pine. It was planted in Concord in Sydney in the 1940s. An outbreak of cypress weevil came in and essentially hammered the streets, and halepensis was removed. Now, having said that, there's a halepensis probably in every suburb in a school, probably 2 or 3 of them, maybe one at the RSL. So there are a few left, but because they're not in high numbers, that selective breeding program of weevils is no longer there. And so we don't end up with the outbreak. So one of the dilemmas that we create when we start losing biodiversity and we start losing genetic diversity within our biodiversity, that is, we've got a very narrow scope, lots of metaceae, and a susceptible plant, is we start to build up the predator or the pathogen quite rapidly. And so we end up with lots of the beetle wanting to do damage, and they do damage. So that's something, again, that we need to think about in terms of urban forest. And also when you're looking at a problem, if you see a problem in a street where there's 30 or 40 of something and you're seeing a similar problem, you'll say, is this a broader spread problem, or is this something that we've created because we've created an environment that is undesirable? So does that answer that question for you, Peter, in terms of...
It does, yeah.
And
I think it's interesting that you talked about taking whatever you call them, suckers of palms. When you propagate bananas, you just cut an eye out, and there's absolutely no roots whatsoever, and it will produce new roots happily.
Yeah. And so we're just doing vegetative propagation. And anytime we do vegetative propagation, we're in terms of overall, we're doing cloning. Now, you do get slight genetic shifts, and that can also be a problem. And the one that's a problem there is that horrible Pyrus caleriana. Love them and hate them at the same time. We have now seen enough cultivars out there where there's been a slight genetic shift and a slight genetic shift, and someone selected a little sport. So a branch has a little mutation at the apical meristem, and it's a little different thereafter, and someone cuts that off. That genetic variance is enough to get away with having enough variation for reproduction. So by itself, the rule is plants don't have sex with themselves. You get told to do it. I get told to do it all the time. Go F yourself. But plants can't do that in general. Most of them won't. There are a few that do, but most plants, they want genetic diversity, so they cross with a different plant, and they have ways of attacking and stopping the pollination from themselves, including opening the pollen and the gynoecium up at different times to functionality. So where we get the problem is, as these clones start to become more and more apparent, is we're starting to see genetic reproduction going on, sexual reproduction. So in Columbus, when I was over there about 4 or 5 years ago, there were thickets of caleriana pairs growing on the hillsides. So be prepared for the caleriana outbreak. It will come. It might be another 10 or 20 years, but it is on the way. So the fact that it doesn't reproduce now does not mean that it's not going to.
Mark, I just sent you through the photos Nat was talking about. For some reason, I couldn't forward you his email, but hopefully you'll get the photos.
Thanks, Peter. I couldn't find it to save myself.
Yeah, it's okay. We'll see how it goes. Okay. It wants me to go to TAFE. I don't want to go to TAFE. Inbox. You said you've just sent it, did you, Peter?
Yeah.
Yeah, it hasn't come through as yet.
Okay.
Okay, so whilst waiting for that to come through, yes, the other thing I was going to say is we use hormonal sprays. So to set a fruit, and this is another part of it, not all fruits will develop properly if they're not fertile. And so if you get an infertile fruit, the secret is to get it to grow. Now they're doing work on mangoes, for example, with an infertile or a knobby seed inside, so no value, just a little thing about the size of a big almond. But then you lose flesh. It's the same with lots of things. So One of the things we do is, for example, on grapes, we spray hormones on them to make the fruit enlarge. Otherwise, it won't. It's not getting the message that it's fertilized, so it stops producing a big fruit. And of course, we want large fruit. So there are problems when you start to get with some of those things. I said getting the right mix together, the right plant, and bananas have been an incredible one. That's a multi-billion-dollar Cavendish is a multi-billion-dollar plant. Hard to think of it like that, but that's what it is. Okay, let's see if it's in. No, it's not.
Maybe it's there's a bit of confusion going on. Nat tried to send some photos with another question. I might.
Okay, if we have problems, Peter, I'll get you to open your screen.
Well, yeah. The problem is that the most recent email I've got from Nat doesn't show the photos he's talking about, so I might have to just have a chat to you, Nat, and round this up for next session.
I'm fine with that.
Yeah, so if I can, I've got it here. I might just flick it through.
Yeah, I think we probably.
I'm happy if you've got them on your screen, Matt. Nat, if you want to, I'll stop sharing and you can share.
Yeah, we could do that.
And I'll talk about it happily.
I'm on the phone, so I don't know how to do that.
Oh, right.
Okay. So I haven't seen them, so very hard to talk about them.
Yeah, we should be able to get to it next session, eh?
You
Sure.
did send them on though, did you, Peter? Yeah, I've just had a message. Oh no, it's not from you.
I've just
Maybe
sent.
it is. Yeah, it's you. I've just got it.
These are the Gumtree photos from. I think we might have talked about these previously. So happy to park this and sort it out.
Can you see this?
Yeah.
Yeah. So what's the concern about the tree, Nat?
Oh, not a real concern. I just thought it was an interesting topic. It was one I wasn't assessing, but I noticed, and it had a Phalaenus infection down the base.
Yeah.
And to a layperson, it might look alarming. So I went and sounded it, and you know, it didn't sound great, but it was an interesting one. I think I might have written in the email. I just nutted out a QTRA. You know, just on in my mind, and I asked the same question with a couple of other colleagues, and we all came up with a slightly different risk of harm. But
okay, the first thing to keep in mind is that, in general, Phalaenus doesn't want things to fail. So it's not something that frequently causes a tree to fail. Not that it doesn't, it's just not frequent. And this one's behaving as a pathogen. You can see here, it's rolling the edges over. And if I look at that tree, I go back here, I think I can make it bigger. So let me make it bigger.
it's actually gotten healthier. I went back and looked at some street views. The decay looks as if it's spread, but the canopy looks healthier than what it was. So
Yeah, look, there's no correlation, many occasions, between Phalaenus and tree health. There are some exceptions to that. So sometimes you'll see it kill Saligna, you'll see it kill Scoparia, Nicolai. But if I look down in Canberra, for example, Eucalyptus globulus, I would expect every second globulus with a trunk of 50 centimeters or more to have Phalaenus in it. I just can't think of not seeing them there. And yes, it does perhaps lead to a slightly higher increase of mortality in some trees, but globulus seems to cope with it really well. But what it is doing is it's stimulating as it loses strength. We get this response down the bottom called thigmomorphogenesis, a lovely little word, where this starts to behave, the wood starts to behave differently, adds more tissue on because we're getting flexing, and it adds stronger tissue on. So you may in fact have no real loss of strength and potentially may have increased strength at the base there. And so the response to that, of course, is we cut it down, which is just beyond me. Now, if that occurred up in a fork in a tree, and I can speak to this, Jessica tells me there were about school 120 this year. I would say that on an average, each school that I go to probably has 2 or 3 cavities in trees. And they're desirable. You know, they've got birds in them, they're nest points, they're not being removed. And I'm not going around seeing a whole pile of failed cavities. So the cavities are desirable. What forms those cavities? Usually Phalaenus. So we have this odd thing that we accept the cavity, but we get really uncomfortable. And I understand it when I see this. What is interesting about this, though, Nat, is see how bad this year's growth is. Can you see that?
Not really.
The Phalaenus sporocarp, nice big lip here. This year the lip's half the size and it doesn't go all the way through. It's struggling.
So that's the fungus struggling, not the tree.
That's it could be the fungal strategy, or it could be what we talk about, and that is Phalaenus is not really good down at the ground level. So other things are more efficient at being saprophytes, and therefore they're going to want to compete against the Phalaenus to take advantage of that material. So Phalaenus up in the trunk does better than Phalaenus down at ground level. Not saying you don't get Phalaenus down at ground level, but it's got to compete now against soil-borne fungi rather than aerial fungi. So different area. Does that make sense?
Yep.
And that's one of the reasons why Phalaenus doesn't want the stem to fail, is that stem fails and hits the ground. First of all, it can't continue to attack new tissue and form the perennial canker that allows it to keep itself alive for years and years and years and years. It can't come into contact with birds that want to help spread it. It can't be spread around as that readily. And now it's got to outcompete everything on the ground. And ground fungi are much more competitive at saprophytic actions. And saprophytic actions are primarily what this fungus is dealing with. It is very weakly pathogenic in most species. Sometimes it gets a little bit stronger, as I said, in those ones that it kills off. But in most cases, it's not a good pathogen or a good saprophyte. It sort of fits in that middle no man's land. So hit the ground where there are lots of saprophytic fungi, and you're at a distinct disadvantage. So why do I want to do that? Now, I'm anthropomorphizing, and I understand that there's not this thinking process, but there is this evolutionary advantage of the fungus regulating what it does so it doesn't fail. Mariano. Hi. So next question. Nat, did that answer your questions on that?
Yeah, I more sort of wasn't a tree I was concerned about. It was one I discussed with a couple of colleagues, and just in terms of QTRA, it was quite a busy road at 2 points in the day and at 60 kilometres an hour, and we just sort of beat it around the bush a bit and came up with, I think one of them came up with 1 in 30,000, and it all, it was interesting to see people's takes on it and their tendency to be more risk averse than others. So,
Mark, did you see that? I guess you didn't. Did you see the email associated with this at all? Hi again, Peter. I was recently assessing trees and noticed a wound at the base of the eucalyptus species adjacent to a busy road with a speed limit of 60 kilometres an hour. On closer inspection of the wounded area, I found Phalaenus fungal bracket. The tree was leaning at approximately 20 degrees towards the busy road. Based on my estimate of vehicle frequency per minute, I chose target 1. I'm assuming this is QTRA. With a size rating of 2, a mass reduction of 100%, and a probability of failure of 4. I came to 1 in 10,000. It seemed like a pretty high 1 in 10,000 seemed like a pretty high risk outcome to me.
Yeah, very, very
Can you
high.
hear me,
I
guys?
wasn't comfortable with that. And then I redid it a couple of times and managed to, bring it well down, but it didn't really sit with my gut, so it just went to, it showed me that, I'd use QTR a fair bit. It was interesting to go through the process just quickly.
I think the
Can we do that now, Nat, with everyone present? Do you mind?
Yeah, that'll be good. Yeah.
probability of failure is the interesting part of this equation. Given what Mark's just said about how Philinus works, and
Yeah.
I
Sorry about that.
remember one risk assessor saying to us, don't think about the bit that's missing, think about the bits that's there. The bit that's there is what holds the tree up, not the hollow.
Mm.
Okay. I apologize to Google for doing that to it. Can you see the QTRA calculator in front of you?
Yep.
So we're talking about a car. I don't know how frequent the road was, but you said 60 kilometers an hour. So 420 vehicles, that's about 600 or 700 an hour during the daytime. That's a pretty busy road.
Yeah, you'll have standing traffic there for a lot of the sort of early to mid morning, and same in the afternoon.
Yeah, this is Canberra, early to mid-morning, an hour or so of heavier traffic that lasts for 5 minutes at a time. You call that peak hour, yeah. And size of part, 450 mils and over, so size part 1. No need to talk about mass reduction. And let me ask you, if you look at the tree, is it closer to a 1, closer to a 7, 7 being an ideal tree, 1 being a tree that you think is going to fail sometime very soon and you're cautious and sealing off the road? Obviously, it's not a 1, but which one is it closer to?
I would say 7.
7.
Yeah, 7. And as I said to you, that Philinus tends not to cause failures. So if you turn around and said, look, it's a 6, I would say I'm comfortable with a 6. Some people go down to 5. A few will go down to a 4. And even as a 4, I would say that is already ALARB. That is as low as reasonably practicable. You've got $100 to spend or $125 to spend on removing the tree. And I would argue that you can't remove a tree like that for $125. That's just madness. So on that basis, you've not got anything that's a concern at all. One of the things that was interesting during the conference this year was that Brian Kane gave a presentation on arborists assessing the likelihood of failure of trees with decay in. And I know Peter and I have already discussed this. I have to say I was more than flabbergasted because he showed data, for example, on a tree with, and I'm going to do off memory, I think it was 11% decay. You still had arborists saying that failure was most probable or possible, which just is horribly overestimating the likelihood of failure.
I think one of the reasons that there is that difficulty is that people look at those words and one person will take a certain meaning from it and another person will take a slightly different meaning from it.
Yeah, I think that's true, but then the publication itself gives you some hints. So imminent failure, something that most arborists will rarely see. Oh, okay, in a year. So that's clearly not a dead branch, for example. And I've seen arborists produce a BTRA or the ISA track basic tree risk assessment that have said dead branch, imminent likelihood of failure. Well, that's clearly not what is meant by imminent. Probable during most normal weather conditions means that it seems almost more likely than not to occur, but closer to that end of the world. Even if we said QTRA 1, 10% of the time. Well, again, that's a pretty rare tree. Possible is still, a big margin for things. And I agree with Peter. You get to this strange range, but probable failure on a tree with essentially no decay, because 11, 12, 15, 20 no decay as far as the trees concerned.
I just can't accept that a description of a tree that most arborists don't see very often or wouldn't see many times in a year is an adequate calibration of.
No, I don't think it's an adequate calibration. I think it gives you some form of calibration that we tend to then want to ignore. So, for example, when we talk about the occupation rates, you've got constant, and I understand what constant is. Someone's there pretty well all the time. So, for example, no school grounds would have an area, if you're using BTRA, that would be constant. The next one is it frequent? Well, I would argue that there are rare spots in rare schools where the occupation rate is frequent, but in most cases not frequent, because frequent infers therefore most of the time. And even if I took that down to, say 20 or 30 hours a week, there's not a spot where most students are present 30 hours a week.
Mark, you keep trying to explain these things with these words with numbers. So my frustration is, why not just use the numbers?
Well, I would say we should. I would say that qualitative risk assessment is incredibly problematic, because when we say low, what do we mean? We mean low compared with other trees. And the problem is with trees is the risk is already very small.
I think
so low is meaningless.
I think it's subjective. It just means different things to different people.
Yeah, it does.
Numbers mean different things to different people, but at least there is some sort of baseline to go back to, some sort of calibration that we can use. 10% means 10%.
Oh yeah, look, I don't argue that at all, Peter. I agree that as far as the world goes, I would have everything done with numbers, as you know.
I just think, an argument for using qualitative risk assessment is that some people aren't good with numbers. So, hey, here's a way of doing it without numbers. But my counter to that is, you don't go into a shop and see something for $30. The shopkeeper doesn't say that's 1.5 lobsters, thanks. Give me 1.5 of those red things you've got in your wallet. You use a number. Yeah,
no, we do. But people are, there are fears of numbers, and there's confusion about numbers. Not everyone. I mean, if I can give you a great example, and you can go watch this on YouTube, the level of innumeracy in Australia and America is appalling. And by numeracy, let me just see, quick show of hands. A quarter. How many minutes in a quarter of an hour? Who would say 25? Okay, you can go and watch at university students being asked this question in America, and they will tell you 25 minutes. How many dimes are in a dollar? Now, I'm not American, and I know the answer straight away is 10. How many weeks are in a year? And the answer is they can't work that out. And that's horrible. And so I think that's a big issue, Peter, that people are scared of and traumatized by numbers. So the one thing I will say for the BTRA, the ISA track system, is that it can be implemented by somebody with some degree of proficiency and not with any degree of precision or certainty or reduced uncertainty, with some degree of competency. And in most cases, it should come back low. It is really hard to get a moderate or a high using BTRA if you follow it.
But I think that the presentation at the conference clearly showed that for whatever reason, whether it's the system or for whatever reason, there's a huge disparity in the sorts of results that people are finding.
horrible level of understanding of anatomy, of physiology, of physics, of basic science, of numbers and everything. It just is ghastly. But it's the reality. And I know that if we looked at the same thing here, I think we went through a report only just 2 blocks ago, and the report said the tree had mycelium in the mulch and needed to be removed. You know, there is such a huge disparity in our knowledge. And I would encourage you all just to continue building on what you've got and realizing how complex the subject we deal with, trees, how complex it is. You know, I'd be lucky to know 0.1% of what there is to know about trees. And, you know, I look at that and I think, how the hell do I operate in this environment? And then I look around me at people that have got actually 3 eyes, drag the knuckles on the back of their hands on the pavement 10 foot behind them. And I'm just preparing a presentation at the moment for a talk in June. And one of the things I looked at was the Aidan Botts coroner's report. And, you know, the evidence given by the arborist that the coroner accepted, every time I read it, it's just enough to make me shudder. And I thought, well, that's because coroners don't, can you believe this? Coroners do not understand risk well. And Peter and I both went to a presentation where a member and a solicitor presented information on some trees, 16 Monterey cypress, Cupressus macrocarpa, that overhung the back of a number of houses. 3 branches had dropped in the previous 5 years. And so the question is, you know, are these trees safe or aren't they safe? You know, is there an unacceptable risk or not?
Well, it astounds me that both arborists that were involved agreed that the trees weren't dangerous, and yet
they agree the trees weren't dangerous, but couldn't say that they were safe or that they provided a low risk or whatever it was, the benchmark. And then the member went out, and this is where it gets really crazy. The member went out, looked at the tree, and they informed themselves. They saw the branches overhung the back fence and it continued to overhang the house. And he was satisfied that they were dangerous. So in spite of 2 arborists saying not, the member still formed that view. Now, what I look at statistically is, I bet you there must have been 50 or 100 branches on each Cupressus macrocarpa. So, okay, yep, 3 out of 16 trees, 1/5, have had a failure in the previous 5 years. That's 1 failure in every 25 years for each tree. Again, this inability and this incapacity to crunch numbers is appalling. You know, what's the likelihood that a particular tree was going to drop a branch the next year? The answer is 1 in 25 based on the historic records. And we don't know whether they failed during normal weather or as a result of storm weather at all. So the ideal thing to do is when 3 of those trees have dropped branches, or maybe 2, we don't know whether it was all from 1 tree even, but it was no more than 3 trees, 3 branches, can't be from any more than 3 trees. So the way you treat the 16 trees is you cut all 16 down. Now, where's the logic in that? The logic is this. Niels, you're up recently at my place. You're a male. I concluded that. I determined that the long hair and the dreadlocks don't tell you, but the beard might. And males responsible for more domestic violence. So we're just going to lock you up now. And that was exactly the mentality of the member in this particular judgment was, well, 3 of the trees have dropped a branch, therefore all the trees must be dangerous. Now, that's absurdity. But it's because we do a really poor job of understanding and communicating risk as a profession. And Peter, that was your comment about the arborists. Both arborists present information that
I think that one of the major issues is the wording of the legislation underpinning it. I think it went along the lines of if a tree was an immediate risk, and there was some discussion about what immediate meant. But
hang on, if a tree's not dangerous, why is there immediate risk?
Yeah, exactly. Exactly.
And this goes back to the point I'm saying is they don't understand and they don't communicate risk well.
When you drill down to the meaning of immediate risk, it means that any hazard would conform to that, because immediate means just it could happen without any intervening event, and you don't know when it is, but it could happen soon. And so any hazard could be categorized as that.
Well,
the problem is that they didn't consider the severity of the risk, like what the risk actually was in terms of, is it a low risk, a medium risk, or a high risk? There was no consideration of that. It was just, does it fall into the meaning of an immediate risk? And it did.
and I agree. Again, legislature is written by the legal profession. So if you look at the New South Wales SEP, and sorry guys aren't in New South Wales, but the Transport and Infrastructure SEP tells us that in a school, if an arborist, AQF level 5 arborist assesses it as a tree and determines that it poses a risk, the tree can be removed. Well, by definition, all trees pose a risk. Yes, the risk from this tree is 1 in 100 billion.
That's right.
No, it's a nothingness. And again, it's just a failure of parties involved to understand and communicate risk. And that goes all the way from the judiciary down. And, you know, when the judiciary and the legal profession are writing these documents, they are showing that they don't understand it. So when you get something like Wong v. Shirt, where Justice McHugh makes his judgment, you know, it's great because he communicates so clearly that he does understand risk. But if I read the Aidan Bott judgment, I sit there and think, wow, I'm not sure that the commissioner gets it. And yet, so the coroner, and yet the coroner is making these decisions about risk every single day of their life. That's the stuff they're dealing with, deaths and fatalities. So, you know, when you see a coroner that struggles with it, and you see arborists struggling with it, it's a little question as to why we come up with the answers we get. And it's always going to happen, unfortunately. But yes, it was interesting to see, Peter, the fact that arborists with essentially no decay still thought trees would fail, even when they were given the tomograph and it showed the extent of decay was limited, so they weren't having to guess anymore. They still thought the trees should essentially be removed because they were problematic. They were, you know, high risk. You need to do something, prune the tree, brace it, whatever. And that arbophobic mindset is common in arboriculture, even in Australia.
I couldn't tell you the number of times that people have asked me, oh, is this a widowmaker? Is this tree a widowmaker? Well, I think that if those trees are widowmakers, they're doing a spectacularly bad job, because where are all the widows?
There are a fair few there. I was thinking this century so far, there's been over 100 tree fatalities.
This century,
yeah. And I don't want anyone to struggle and worry about it, but this century there's been close to 1,000 bed fatalities. So, when I say good night to you guys, please get your futon, put it on the ground. Be aware that your days are numbered, because beds kill 2.5 times more people than trees every year. But I still don't know the name of the profession that inspects beds. One day I'll find that out. I'm sure there's a government department that does. Having said that, in a very jocular form, there are some things I understand that bias that age is one of them, young children and elderly people that fall off beds. And I say it with the grace of love for my wife. I hope that's how she dies. I hope she rolls out of her bed one night and kills herself. It would just make such a great story when I'm talking to arborists to say, hey, look, you know, there was no news media outside the front of my place. No one give a damn. But when a tree falls over and squashes somebody, they usually care, and they really care. There's 2 times when they really care: if it's a child or if they're cute. Okay, so a poor guy in Collie a number of years ago was hit by a tree. Only 2 mentions in all of the media in Australia of this poor guy getting killed by a tree. Bridget Wright gets hit and bang, it's all over the media, all over the world. You know, it's the cuteness value. So what happens is if I get hit by a tree, none of you will ever know, okay? Because there'll be zero media reporting on it.
We might hear about it through the grapevine.
I was called by a journalist near Canberra, ABC, and we had a storm come through, and he wanted to talk to me as an arborist about, you know, trees and the dramas and all this stuff. And I said, yeah, but what I'll be talking more from the fact that these are really rare. And he obviously didn't like what I said because he never called me back. I wasn't dramatic enough for him.
Yeah, look,
Because he wanted to, you know, video me in front of a fallen tree. So no, I'm not being part of that.
yeah. Let me. You can video me in front of a tree with all sorts of problems and features in it that make people shudder, and you'll see me there without a helmet on and without fear of the tree falling on me. You know, it's just, it's induced in our thinking processes, and it's very hard to get out of it, and it's pervasive through society. And one of the things that I've said before that I always find fascinating, when I say, look, each year on average in Australia at the moment, about 5 people a year are killed by trees. And that compares with about 1,200 from motor vehicles. It doesn't take a lot for people to realise that the risk from trees is incredibly small. And most people respond with, is that all? And you say, yeah, that's it. You can go read on it. You know, there's data there. We make it all available. You know, that's such a small number that I wonder how we get away with. And just doing the calculations on it today, tree risk assessment, formal tree risk assessment in Australia is an almost $500 million a year income industry. That's amazing. You know, could you imagine 10 million being spent on bed inspections? When we have a specialty tree risk company selling at over $10 million, it gives
Surely
you
it's more the person in the bed than the bed.
Sorry?
Surely it's the person on the bed and what they're doing with the bed rather than the bed sort of collapsing or flipping over and crushing them, though, right?
Yeah, but will they fall from or onto the bed is the classification on ICD-10.
Oh, okay, yeah.
And yes, look,
Jeez,
there's no
that's
doubt it's
amazing.
the person, but you know, when 80% of all tree-related deaths are people going out during storms, you know, can I be honest and say that some of that maybe is the people too? And you know, we're looking at a death a year. What would we do to prevent it? The answer is nothing. We don't prevent sharks. We don't prevent snakes.
Well, we do. We try to prevent sharks, and we actually make it a lot worse by netting and all the rest of the stuff that they do. It's anyway. I won't go into that.
And Peter, the same thing that's called an unintended consequence. Very essence of what I'm talking about at the moment in terms of the presentation. And the unintended consequence of trees, of course, is we hurt more arborists than are killed by trees falling on people during calm, still days. So we transfer the risk that's almost non-existent onto arborists and make it existent. You know, it's fair that an arborist should die to stop somebody from not dying, even though nobody was going to die. That makes, you know, stupidity. But that's what we do. And
I wrote that in a report the other day.
Yeah. And so that's the first thing we do. Second thing is we lose canopy. And I'm just at the numbers crunching stage, so I'll tell you what it is next week if you like. But the loss of canopy in New South Wales schools, we have some fairly good estimates now. And it's horrendous. I mean, the harm that we've done to people from removing trees in the name of risk reduction is actually the opposite. We've hurt more people and done more harm than we've done good by getting rid of the trees. You know, in a fair and equal world, about every 30 or 40 years, some kid should get killed in a school by a falling tree.
I think the problem is that the number of people that we've killed by removing trees in schools can only be determined statistically. There's no bleeding body lying on the ground that we can take photographs of.
Well, not in the school, but we can talk about the things that happen. So we know that the health benefits of trees are incredible. We're starting to get a better idea of that now. And we can put a value on that because we put a value on human life when they die, the value of statistical life. So yeah, I think we'll see, and I'll give you the answers next week if you're at WAR. And I'll tell you exactly what the annual harm is now after the removal of thousands and thousands of trees. The number of trees estimated to be removed in New South Wales schools is about 45,000 trees since Bridget Wright died, and at least half of those were healthy but had structural issues. That's pretty scary when you think about it.
The other thing that I find disturbing is that it's very difficult to get new trees planted in schools. They'll quickly remove them, but they won't replant.
Yeah, it's interesting. Again, our SEP requires that they replant. So, that means they won't be. So the law says you've got to do it. It doesn't apply when the government's involved, and it doesn't apply even though the SEP says that the tree can be removed and must be replanted. And in fact, I cop abuse when I say, no, you can't remove that tree. You'll need to apply to a council, and the council can't reasonably refuse it. But if the tree doesn't pose a real and perceivable risk, they have every right to refuse it. I get told to shut up, mind my own business, and they're going to go ahead and remove it anyhow. And that tends to be the protocol. And let's be honest, they're doing it because they're afraid. They're not doing it because they hate trees. They're doing it because they think they're doing the right thing.
The easiest thing I try to tell people after a storm or something, and they say, oh, there's trees down everywhere. And I'm like, look, just walk down your own street, do a count. How many failed, how many didn't? Drive from one side of the city to the other. How many didn't fail? Sure. You might have seen 5 on your way to work, but how many did you see on the way to work that didn't fail, didn't even lose a branch? I mean, you can't say it any more simpler than that. Surely.
Yeah, I agree.